welcome everybody to the December webinar for the Cancer Genomics Cloud. We are doing a year in review. We typically don't do a December webinar, but this year, this seemed like a good opportunity to actually spend some time talking about what we've done this year. My name is Sarah Fisher. I am a community engagement manager on the Cancer Genomics Cloud. I have a background in evolutionary biology, and I've worked with the CGC for the last 2 years. And I think several people may know me from office hours or just from getting emails from me. And I would like to introduce my partner in cloud genomics, Rowan Beck.
Hi, everybody. As Sarah wonderfully introduced, my name is Rowan. I have a background in genetics and toxicology and kind of public health. I've worked with the Cancer Genomics Cloud now for almost 3 years, so happy to be here and happy to be hosting this December webinar where we get to showcase some of the great, projects and tools and all sorts of excellent items that you guys have helped us put on the CGC. So, with that said, I guess we should just keep going. So there are a few items we're going to talk about today. You can see them listed here. Essentially, we're just going to talk about some of the features on the platform. These include public projects and tools, new integrations to make it easier for you to get your data. Then we'll focus on more community-centered events. So these include like events and conferences, teaching collaborations, as well as the accomplishments of our CGC community. So to kick us off, we're going to be talking about some of our new public projects.
Before you dive too far in, Rowan, I do want to let everybody know if you are using, if you're not familiar with Teams, there is a Q&A button that you should be able to click. If at any time you have any questions or want to ask us to focus on anything, please feel free to add that into the Q&A. And we also have a couple of questions we're interested in you answering. For example, what are you most excited to see on the CGC in the next year? And what is your preferred method of learning? Webinars, documentation, workshops? Please go ahead and throw any questions you have in there. And now, Rowan, my apologies, I'll let you take it away.
It's quite all right. I was just excited to showcase some of these new projects. So to begin with, we will talk about some public projects that we've introduced on the CGC in 2024. And you'll notice many of these projects are centered around imaging. So we really tried to enhance our imaging capabilities on the CGC. And we did so through the introduction of several different new tools, which are featured in our public projects. So the first one is UMetaFlow, and this is for a metabolomics workflow. It is for data processing and analysis. So it's written in Python. You'll find this on the CGC, same place you'll find all of our other public projects by navigating along that top blue banner and selecting public projects. Then once you copy this over, you'll find all of the data analysis in our Data Studio section. So this one is written in Python, and it offers step-by-step demonstrations and examples of how to use it. It goes through 4 different notebooks here for pre-processing, quantification, export, and another type of export. And it allows you to compute, explore the data, visualize, as well as test out new analytical steps and processes. So again, you can find this on our public projects page.
Yeah, and I'm also going to throw the link into the chat.
Thank you, Sarah.
You will have to use the link, but since it's hard to click a link in a presentation, and these slides will also be available once we're done. So yeah, this seems like a very exciting project to have, like a good metabolomics workflow that is just like point and click.
Point and click. I like that it's separated into 4 different components, so you don't get bogged down with a piece of code or script that's, like 200 pages long. Here, you can just take it one step at a time and troubleshoot each one step and ensure that it works before moving on to the next step.
Cool.
So our next project is for, hold on, let me click on this link, PyImageJ. So
I
this
was very
is a
excited
new
about this.
integration we have. Yeah, you guys have probably heard of ImageJ. Well, we have that ImageJ now through Python on our Data Studio. So this integrates ImageJ with Python's ecosystem. So it has the features you're familiar with in ImageJ, but now you can combine them with Python tools like NumPy, SciPy, and other packages on Python. So again, this is in our Data Studio from our public projects page. And there is a demo notebook that was developed as kind of an introduction to using PyImageJ. And you'll find this under the interactive analysis section of the public project. So there's a screenshot on the right. It just shows you kind of a description of what this project entails and how to copy this project so that you kind of own it and you can modify it and test out the scripts as needed. So this was a collaboration, and we have some of the information here on the bottom left, so you can actually read more about ImageJ and this new implementation of PyImageJ, as well as we have, once again, the link on the very bottom, which I'm sure Sarah will put into the chat.
Sure
But
did.
this is a really great interactive tool. You can kind of, because it's a Python notebook, you can really modify in real time some of these plots and visualize and explore the data in new ways that you're not, normally able to do. So I
One of
think
the
this is a
things,
great. Oh, sorry, go ahead.
yeah, no. One of the things that I was so excited about with having PyImageJ available now is everybody's really interested in doing AI and machine learning with imaging now, right? And one of the things that sometimes people end up giving short shrift to or not paying enough attention to is how much pre-processing you may need to do with images before you can actually feed them into your machine learning algorithms. So PyImageJ is a Swiss Army knife for images. And so having this available, I think, can really help some people kickstart some cool projects.
Definitely. And we have documentation as well as just honestly online. PyImageJ has plenty of documentation with tutorials and example scripts. So it's really easy to get started. So utilizing this with some of the image data that we host on the CGC really makes it a great combination, and it makes it much easier to analyze this data. And for our next public project, we are featuring a tool called Vitess. So this public project was actually created by our colleague Phil, and it is featuring a tool called Vitess. And this is used for visualizing and analyzing single cell data specifically. So this is really a great tool for interactively working with the data. So you're able to visualize things, but not only visualize, you're actually able to make modifications in real time as well. So you'll see in the screenshot here along the left panel, you'll see different layer controls. So you can change the segmentation, you can change colors in real time, you can expand or kind of zoom in on your visualization and look at a specific clustering of cells. You can look for specific expression. There's a lot you can do, and this is all available in our public project. It supports several different data types, so it makes it really easy to work with CSVs, JSON files, OM, TIFF, and more.
Vitess is one of the things that our engineers developed directly from user request. We have, through working with HTAN, which we'll hear more about later, we have MC Micro, which is an end-to-end pipeline for whole slide imaging. That's really useful for the spatial transcriptomics of single cell sequencing. But it doesn't have as much of the sort of interactive component. And Vitess can take some of the outputs of MC Micro and let you interact with it directly in a Jupyter notebook. It's just one of many ways that, Rowan, you say this all the time, that the Cancer Genomics Cloud can be as simple or as complex as you want it to be, as complex as you're willing to let it get. MC Micro is very easy to use for your whole slide imaging. Being able to add the powers of Vitesse to that output, I think is a pretty
easy,
cool
honestly.
tool. Yeah.
Yeah. For someone like me, honestly, who did not go and do a PhD with single cell data, it's really great to be able to take a scatter plot or a t-SNE plot and highlight certain clusters or portions, that have expression that I'm interested in, so that I don't have to sort through all of the data. I can specifically gather a group of cells and know that, these are where I'm interested in looking. It makes it just click in a different kind of way. And I think that's really useful for those of us who are visual learners.
Cool.
All right, so our next section is going to be more on featured tools. Keep in mind, there are plenty of public projects. We just featured these 3 because we did not want to spend the entire webinar talking about public projects, but please do check them out. They're available, again, on the platform. Just select public projects from that blue banner. There are several that you can list. They have descriptions. They are all of the data that you will need is provided within the project. So that makes it really a useful tool for learning. So if you're just curious about learning a new tool like Vitesse or PyImageJ, just copy that project. Everything you need is ready to go. The scripts are there. Everything is commented out, and it should be a really interactive, easy way to learn. So as I mentioned, our next section is featured tools, and we're going to start off with a recent collaboration, and this is called 3D VisSnip. So this is a tool that helps to visualize large 3-dimensional structures. So it uses ICN3D, which is a web-based 3D viewer, and it represents molecular structures. So this is a little more advanced than your average old school kind of chemistry visualization where it was just sticks and zeros. Here you can actually interact with the molecule in 3D. You can highlight specific portions. There are many different options you can use to really visualize and interact with the molecule. So 3D VisSnip exists to extract the missense mutations. It reads VCF files, so you can just upload the VCF file. And it returns a table that is interactive. So you can sort through it, you can filter through it, and it provides you all the information you need, like gene ID and other information. And not only from there, you can actually interact with it. Like I said, you could visualize it, you can modify it. You'll find this on our platform, according to the screenshots here, if you select public apps and then navigate to interactive web apps. This is built in R-Shiny. So you'll be taken to a kind of new page where you can create a new instance to run this tool. So we do have a video of it here. I don't necessarily need to play the entire video, but just to sort you through it, this is what you see when you first load 3D VisSnip. If you click the Visualize tab, you're able to upload a VCF and then interact with the molecule in real time. You'll see the table with the variants listed along the bottom here. And again, you can sort through it. You can highlight which ones are potentially of interest to you, whether they're harmful or mutagenic or so forth. So this was a collaboration with our NCI CBIT team. So this is actually originally on the NCI website, and they wanted to reach an additional user base. So we were able to take this and put it on the CGC as well to kind of reach a different audience. So this was built in collaboration with the team at the Biomedical Informatics and Information Technology Center, as well as our CGC excellent engineers who were able to get this on our platform. So this is definitely something worth checking out. If you don't have a VCF file, do not worry. It actually comes with one, so you don't have to panic. You can just test it out with the example file that's provided, and you can get familiar with it that way before moving on to your own data.
I've heard you talk about 3D VisSnip many times, but this is the first time I really understood that you're actually starting from a variant call file, from a VCF file, instead of starting from, I don't really know much about proteomics. So I sort of assumed that this was going to be starting from some file format that was just for protein viewing, but it actually already has the ICN 3D protein structures incorporated, and it's just going to actually show you on that structure what the variants do. That's really cool.
It is great, and it's honestly, there's so much documentation and tutorials for utilizing this tool that it makes it really easy, even if you're a little bit hesitant to jump into something new and you don't have experience working with this. It's pretty interactive. It's intuitive. You can really just click around and explore. You're not going to mess anything up. And like I said, there is an example file, so you don't have to worry about finding the data to get started. You can just kind of try it out and see,
That's
and then
awesome.
later on replace that. Yeah. So that is one of our latest collaborations with this CBIT team. We do have some other items from them as well, like Omics Circos for visualizing omics data in a Circos plot. We also have MOPA is a multi-omics kind of workflow that is available on the CGC as well. And these make it really easy to analyze complex data in a way that you can visualize and just quickly kind of get results for. So do check those out. We have public projects for each of those, as well as these interactive RShiny apps. You can also reach out to us if you have any questions. We're happy to walk you through it or point you toward videos or documentation, whatever it is you need. So our next item is Cell Ranger. So I know people have been
I
waiting
was so
for
excited.
this. Well, the Cell Ranger, for those of you who aren't informed about Cell Ranger, like I wasn't sure how our implementation would be, but it's actually really a fantastic implementation on the CGC. So this is an analysis pipeline that it demultiplexes samples. It does barcode processing, gene counting, and so forth. It does many different things in one. So because of this, we have split it up into 4 different tools. One is the Cell Ranger aggregator. The other one is count. We have reanalyze, and then we have VDJ. So this does sequence assembly. So this paired with access to dbGaP or SRA data really just opens up a world of possibilities. Now you can access all your favorite data sets and you can analyze them with Cell Ranger all in the same space. So we have had users asking for this, and we now have it available to our community. So we're really excited to put that out there. I don't know if Sarah, you have any experience using Cell Ranger.
Yeah, I've played around with this. I've only used it with the public data sets that 10x Genomics puts out on their website. But I like for a long time, single cell sequencing has felt like a mysterious country to me. Because I came of age when we were all very excited about bulk RNA sequencing.
Same.
So all this newfangled single cell, it felt intimidating to me. But I was able to working with toy data sets, which admittedly toy data sets tend to be well behaved. I was able to work with Cell Ranger count pretty easily. One of the really lovely things that this implementation does is it outputs tons of different files for different uses. But one file type it outloads is a .cloop file, which is for the 10x Genomics Loop browser. And so you can run your data through Cell Ranger count and then download this really very small file. And directly on your computer, work with the Loop browser to investigate your clusters and even do reclustering. I was really pleased with like how easy this was to use. Now, granted, it's going to be easier to use with 10x Genomics data rather than any of the other sort of library preparations. But if you've got 10x data, it can be analyzed on the CGC. And as Rowan said, like we also have a lot of different ways that you can connect directly to the sequence read archive, the NCBI's SRA. And so you can pull in and reanalyze single cell data from the SRA in a matter of hours rather than the days that it usually takes me to like wrangle data down and get it all situated before I can start analyzing.
Not only hours, but you said you get an output of just this tiny little file. And I've heard you talk about it before in the Loop browser. So that definitely sounds like something we should do a tutorial video on to
Yeah,
just show people kind of what the output is and kind of how to interact with it, since this is relatively new on our platform.
yeah, cool.
But great, yeah. So moving on, uh, I know you guys are already familiar with Data Studio, but I did want to showcase some of our newer Data Studio offerings. So these on the CGC are the OHIF Image Viewer and SAS Studio. So both of these are relatively recent additions to the CGC. They're joining Python and RStudio. With the OHIF Image Viewer, you're able to visualize images in stacks and interact with them. You can label, you can leave markings, things like that. You can visualize things. Not only that, sorry, again, inarticulate, but you can
You're
interact
doing
with these in
great.
a way that you normally can with images. So you can change the contrast, you can highlight certain regions, and you can play everything back to you in whatever speed you need as well. So it's kind of hard to describe, but there really is a lot you can do with the OHIF image viewer. We do have tutorial videos and documentation
Mhm.
available so that you can actually try it out yourself with some toy data. And then our other offering is SAS Studio. So all the fun of SAS, and now it's on the CGC. So you can connect SAS to your favorite data sets, whether it's accessing them through dbGaP or your own private data that you've uploaded. You can very easily, it's connected to your project, so you can just have access to that data right off the bat and run all your SAS scripts as usual. You can visualize them within the Data Studio and save just the same way you'd normally be able to do. And in addition to these two Data Studio analysis tools, we actually do have custom environments available for RStudio and Python. These are feature flagged, so you'll need to request them. But for those of you who aren't sure what this custom environment means, you can load whatever packages you need in R or Python, get everything ready for your analysis, and then you can save that environment so that the next time you need to complete your analysis, you don't have to start from the beginning and reload all of your packages. So this will save considerable amount of time in some instances. I know there are, RStudio packages, for example, that take a very long time to load because they have many dependencies. So, for these use cases, it makes sense and makes it a lot easier than to just resume your analysis without having to worry about starting over.
Yeah, the custom environments is probably one of our previously most requested features,
Mhm.
simply because the Data Studio environment on the CGC, which is our interactive scripting environment, for the most part works exactly as you expect it to. Like RStudio or the Jupyter notebooks, when you boot them up, it feels just like using it on your local computer,
Mhm.
except for when you install libraries, it doesn't, it used to not stay. You would have to reinstall the libraries again. Um, that was for reasons that made sense when we developed it, because you're building stuff into a virtual machine, but as Rowan said, it could be pretty inconvenient. So this feature is still in beta, which is why Rowan has put the little feature flag there. But it's pretty easy to get it turned on for you. So just be in touch.
Yeah, just notify us or support and we can get that for you. I've tested it out myself and actually I've had really good results with it. So it's made it a lot more convenient to complete my analysis without having to worry about, again, starting over and reloading some of the things. So just little things like that we're trying to make easier so that it's a more enjoyable experience to just do your analysis, complete your analysis, pause, start again, whatever it takes. Okay, moving on to our next section, we have new integrations. So these are some of our newer integrations with some other data commons resources. So I'm going to start us out with just talking about the ICDC. So The ICDC stands for the Integrated Canine Data Commons. And this is established essentially to do research on human cancers through comparative analysis with canine cancer. So there are a lot of similarities there. So they use canine cancer to understand the molecular kind of underlyings of cancer and then translate that. The data in ICDC is sourced from multiple different programs and projects. Again, it's focused on canine subjects. It's harmonized and it's in an integrated data model. So it's really very easy to navigate through the ICDC. It's very easy to then take that data and import it into the CGC. So you would do the same as usual, where you're kind of navigating around and creating a cohort, selecting data that you're interested in and adding it to your cart, just like on Amazon. From there, you'll go to that cart page, and you can either download a manifest to upload on the CGC, or there is a button, which you can see on this bottom right picture here, that just says export to the Cancer Genomics Cloud. And if you select this button, it'll ask you to log into the CGC. And then from there, you'll see a page that just says, which project do you want to put this in? So you can create a new project, or you can use an existing project, and you'll then be taken to your Files tab, where you'll see all these new files available to you. So you can learn more by following the link here at the very bottom, caninecommons.cancer.gov. If you'd like to learn more, we also have a webinar, a 2-part webinar series that we completed earlier this year. Sarah, do you want to jump in while I take a sip of water?
I do. So we ran this, I called it a mini symposium. It was a 2-part webinar series where we had our headliner, Dr. Elaine Ostrander, who works at, I'm forgetting her title, but it's an important one at the NHGRI. And I'm looking it up because it's worth getting it right. Elaine,
this was a great webinar, though.
It was a fantastic webinar. She's the Chief and NIH Distinguished Investigator of the Cancer Genetics and Comparative Genomics branch at NHGRI. And then we also had Dr. Heather Gardner, Dr. Cheryl London, Dr. Shaying Zhao, and Gina Kuffel, who is a technical program manager at the ICDC, talking about canine cancer. Before this webinar, would often tell people, don't sleep on the canine data because it's really useful for teaching purposes. Canine data does not have the same kind of personal identifying information that we're concerned about with human data. And so if you're in a teaching context, you can get all of this great data out of the ICDC to use to teach people how to analyze cancer genomics and transcriptomics data. That was what I thought was cool about it before this webinar series. In the webinar series, I learned that dogs get cancer in very similar ways to humans. In fact, the incident of spontaneous cancer in dogs is possibly 5 times that of humans. Plus, dogs tend to be in the same environments that humans are. So understanding the environmental and genetic components of canine cancer helps human cancer a lot. And also, we love our dogs and we want them to be well. These talks in this webinar series were fantastic. And the work that's been done on creating this integrated canine data commons is also incredible. And I'm very happy that we've been able to integrate so well with this other component of the cancer research data commons.
Absolutely. For anyone interested, really do check out these webinars. As Sarah mentioned, I really didn't appreciate to what extent that environmental kind of variable would play, and how it made perfect sense to study canine cancers in this context. So I learned a lot through this webinar. It was great. There was discussion. It was just fantastic. So again, we have part 1 and part 2 available. So please do check it out. This is just one of our integrations, but it is a fantastic one. So moving forward, we also have the CTDC, Clinical and Translational Data Commons. So this is our newest data commons integration. There are many different data types here: clinical, molecular, sequencing data, all sorts. They are harmonized across NCI's Cancer Data Standards Registry and Repository Commons Data Elements. This is why we have all the acronyms. But yeah, there's a lot of data here that's available. This is very new, so it's still being developed. They're still making additions to this. But again, this is a great management system. You can go in and select data that you're interested in, add it to your cart, and once again, you can either download a manifest or directly export it to the CGC. It makes it really easy. There's a link available for you. Again, you'll just be asked to log into the CGC, and all of the data that you're interested in will be deposited. The symbolic links will be made available within your project, so you can work with it. So I haven't had too much of a chance actually to really work with some of the data available here, but I do know that they have a great graphical user interface, the GUI. Not only that, but you can access the data through an API. So it makes it really easy if you're already using that API to kind of move data around, modify it. We also have the 7 Bridges command line interface and API resources available. So really, once you're in that space, it's just very easy to integrate this data with the CGC. Again, you can learn more by visiting the link at the bottom here, clinical.datacommons.cancer.gov. And then for the last integration we're going to be talking about today, we have many more, keep in mind, but for today's, this is the CDS, the Cancer Data Service. So this provides data storage and sharing capabilities for NCI-funded studies that are not falling under other categories, essentially. So these don't match an existing data commons category or fit into current criteria for. So often these are just smaller studies, smaller, they can still be thousands of samples large, but you know, it's not the TCGA, it's something else.
Right.
So it's really worth it to browse through and find something here because they host just a wealth of data, including the Human Tumor Atlas Network, HTAM. They have the Division of Cancer Control and Population Scientists, Childhood Cancer Data Initiative, CCDI, as well as independent research projects. And once again, super easy to navigate through here. You can select whichever files are of interest to you and add them to your cart. From here, download a manifest, or once again, click the Export to Cancer Genomics Cloud button. You'll be asked to log in, and all of the files will be made available to you. So you can then use any of our tools or Data Studio resources to analyze the data right in your project space. So these are only a few of our cloud data commons resources. Sorry.
You got it. You got
yeah.
there.
Yeah, if you are interested in collaborating with us, please let us know. We're always looking to make it easier for our community to kind of work together and integrate data with tools. So
yeah, one of the things that I do want to just spend a second on is the fact that the way that these things have been integrated with in the Cancer Research Data Commons, if you have access to data and load that data into a workspace on the Cancer Genomics Cloud, you're not having to pay for the storage for that data. This is really sort of getting us to that promise of the Cancer Genomics Cloud where you're analyzing the data where it lives. The data sets are big. We don't want to have to move them around all the time. Here, you're actually able to connect to where the data exists and do your analysis on it without having to download it. And that's something that sometimes we forget to kind of mention. But it's a big part of why the CGC does what it does.
Yeah, it saves time, it saves storage. You can mix and match as well. So, you know, if you have data in your project from one of these resources, you can also upload your own data and analyze them together. So it's not either or anything like that. Really, you're free to do your analysis however you need to do it. So keep that in mind. We'll be moving on to the next section, conferences and events. And Sarah, we're going to start with the AIDR, the AI Data Readiness Challenge, if you'd like to talk a little bit about it.
Yeah, this was a really interesting thing for, that we provided support for. We did not run this challenge, but this was through challenge.gov. So this was one of those competitive data crunching challenges that the NCI put on. And the purpose of it was to sort of probe to see how ready for AI usage the data provided in the Cancer Research Data Commons was. We had 49 teams participate, a total of 145 participants, and they used various aspects of the CRDC and attempted to take the data, aggregate it, and build just a simple machine learning model. It was a really interesting experience for me. And one of the participants, Agnes McFarlane, spoke about her experiences at the CRDC symposium. And so we've got a link here, and I'll pop it into the chat in just a second, where you can watch what she said about participating in this challenge.
Yep, it was our first time doing something to this extent with the data readiness, but I'm sure we'll be doing more along the lines of kind of assessing the data, as Sarah mentioned, in the future. So, keep on the lookout for that. We'll post, on our website, social media, things like that, and the nci.gov. I'm sure you can find information as well. One of the other events that we participated in this year was AACR, so the American Association for Cancer Research, and this was a conference held earlier this year in San Diego, California. Here we presented 2 posters as well as a talk. And the talk was about leveraging real-world pancreatic cancer datasets to drive drug discovery. So it was really focused on drug discovery and patient health among the PanCAN network. So that's the Pancreatic Cancer Action Network. We also had 2 posters. So one was CGC-hosted comprehensive pipelines for space. So it was a spatial transcriptomic analysis kind of overview. And the second poster was with our CGC collaborator, Vardui. And this was about this algorithm, Connect the Dots, CTD2. And it essentially just looks at disease genes in complex networks. So it's a weighted gene co-network analysis. I think I got that abbreviation correct in my mind. It's really complex stuff, but we've made it available to you on the CGC, so you don't actually even need to do the math behind it all. It's really beautiful.
Yes, and Vardui has promised me that she's happy to tell people how to make the adjacency matrix that they need that feeds into CTD2. So CTD2 is actually not quite available yet, Rowan. CTD2 is a considerable upgrade from CTD. CTD is available. The newer CTD2 is basically, they wrote the first one in R, and they've written this one in Python, and Python is so much faster and so much more lightweight. And so look for that to be finalized sometime in 2025.
Yeah, if you're interested in learning more, we actually have a webinar on this algorithm as well. So we can pop that in the chat at some point. But it's cancergenomicscloud.org/webinars, and you can find all our previous webinars, including a talk on CTD. I've also included some photos here on the left of members of the CGC team and Valsera, just having a lot of fun at AACR and getting to interact with some of our user community. So another conference we got to participate in or with is HTAN. So HTAN stands for the Human Tumor Atlas Network, and this is a collection of data. It's kind of imaging, spatial, transcriptomics, all sorts of different types of data. And it promotes the creation of different pipelines, visualization techniques, algorithms for the purpose of addressing cancer-focused questions using this HTAN data. So we had our second annual data jamboree just earlier in November. And the first one we had was virtual. So it was kind of last year's, it was our attempt at just trying something new. And it was so successful that we were able to do this now in person just a few weeks ago. In total, there were 45 participants split into 6 teams. And you can see that there was a variety in kind of what each project was about. Here we have a quote also from a participant that I like to put out there, just that the ASHAN Jamboree was an exceptional experience. It brought together people with similar backgrounds to collaborate intensively on a shared project within a limited time frame. This was the most impactful meeting I've ever attended, and I gained valuable scientific insights here. And we had numerous people telling us that they really liked this data jamboree because it was not competitive. So the goal was not to win the data jamboree. The idea was just to work with other people who liked science to do something cool using HTAN data, and they were able to accomplish that. So you'll see a photo on the very bottom here of all of the participants trying to crowd into a room. It was a 3-day event with it was busy, just every day, every hour, someone was doing something, with lots of breaks to present, focus on things that were slowing the groups down, things that were working well. You'll notice the photo on the top here is everyone outside. So one of the things that could or maybe slowed them down slightly was that the fire alarm went off one of the days. So actually, instead of just kind of leaving, everyone took their laptop and ran outside because we still had Wi-Fi outside. So everyone was able to complete their analyses and continue working as if nothing happened, just outside. So you'll see different groups scattered around in that image on the top there, and me walking around kind of making sure everyone was okay and able to continue their work. And then the last item in this category here is the Cancer Research Data Commons 10-year symposium. So I'm sure many of you attended this symposium. It was fantastic. It was a 2-day long symposium that happened in October, and it featured a lot of different success stories highlighting the impact of the CRDC, whether it's data or resources, how those influenced scientific discovery and supported studies to advance cancer treatments. So it highlighted that AIDR challenge again, and you can listen to Agnes's talk by following the link there. Highlighted a lot of the different initiatives, and you'll see the CGC mentioned on many of the slides in many of the talks. So whether it's members of the CGC community who were featured or some of the teaching that we have done, there's lots of different features that were focused on and touched on throughout the entire symposium. So speaking of teaching, that is our next section. So in 2024, we actually had a lot of opportunities to work directly with classrooms. I'm going to jump right into the first one, which is with Purdue. Sarah, do you want to take this one away?
I would love to. So we've worked with Dr. Landman at Purdue for a few years. And this was the first time that I got to go in person to do this workshop. And they run an RNA-seq boot camp, basically, in the summer, mostly for senior PhD students, postdocs, and analysts. So it is pretty high level education, and they teach them all of the fundamentals of doing RNA-seq. And then at the very end, we get to show them the CGC. And sometimes there's a little bit of, oh, why did I have to learn all that coding? But not really. They enjoy getting to learn the coding too. This was really, really neat for me to come, and I taught 3 different classes, 3 different sections of the workshop that day. And in the bottom picture there, the best thing that ever happens when you're teaching is you show one person how to do something, and then you stand back and watch them teach other people. And that was a really fun moment.
I'm sure that was really satisfying being able to do this in person for the first time.
Yeah.
So we do a lot of classes virtually, but something about being there in person, it makes it easier sometimes to answer questions, to just kind of talk things through, whether you're sketching it out on a notepad or just, high level concepts. One of our other classes that we've taught is with Georgetown professor Dr. Yuri Gusev. So we have worked with Yuri for many years now, actually, and we teach a class in the fall as well as in the spring. Primarily, we focus on teaching the Masters in Health Informatics and Data Science program that he hosts at Georgetown. And what's really great about his classes is they've evolved. So he started out just doing really simple items on the CGC, simple analyses. And he is always open to kind of pushing it and seeing what else we can do as we integrate newer items. That's what he wants to teach in the class, which makes perfect sense. So we have gone on to then do single cell RNA-seq. We've done machine learning and imaging analysis. Yeah, again, his latest class used Python to teach students techniques in machine learning and classification, which is like really a cool concept to learn. And being able to do that on the CGC makes it just really easy for students to then have homework that they work on later on and collaborate together. Here we have a couple screenshots. So we have taught these classes virtually, and we've had so much success that students from previous years who have already graduated have come back to say hi or sit in on some of these lectures as well. So a really great collaboration that we're excited to continue. And then our last featured teaching collaboration is with Dr. Min Zhang, and she's a professor of epidemiology and biostatistics at UCI. And essentially, she first worked with us to design a 4-part lecture series to teach undergraduates and graduate students how to run kind of RNA-seq. So this includes both bulk and single cell. It was kind of just an introduction to like data analysis using the CGC, and she invited everyone who was interested. Seeing the success of this, she actually turned it into an NCI-funded R25 initiative called Big Data Training for Cancer Research. And it uses the CGC to access, share, and analyze different types of data sets. And these are all multi-omic kind of data sets. It's a really intense kind of 2-week course she does in the summer. It's open to undergraduates, graduate students, postdocs, really anyone who's interested, professors. And we have done it both virtually as well as we were able to send someone to the university to be there in person as well. So this is a really great collaboration. Again, it leads to a lot of productive collaborations further on as well. We've had several of her students come to us afterwards with ideas for projects that we're able to help fund and really put out there. So
yeah, in
thank
fact,
you.
January's webinar will be
Yes.
given by one of Min's former Big Care students who then started a collaborative project on the CGC and has produced a workflow and is getting ready to actually process a ton of data. So
how cool is that?
It's cool. It's cool.
That's so great to
see the community grow like that.
Yeah, the progress, the community growth, everything. So if you guys are interested in utilizing the CGs for your classes, you can just send us an email. You can visit the website as well, cancergenomicscloud.org/CGC-teaching. And that has some of the links and resources that you'll need. But we're open to working and collaborating with a variety of different types of classes. We have different options that we can do. So really don't be shy. If you know a professor or a teacher who is interested in learning about cloud computing, share this with them as well. We're happy to get in touch, or you can feel free to get in touch with us. And that leads us to our last section of publications and collaborations. How exciting. So earlier this year, yeah, it's still 2024. Earlier this year in March, we had a 4-part invited series that was published highlighting CRDC accomplishments from the past 10 years. So this was a really nice publication to then lead into the 10-year symposium later that year. So it was divided into 4 sections here: lessons learned in future state, resources to share key data or cancer data, cloud-based analytical resources, and this is where we got to contribute to this publication at the CGC. And then finally, core standards and services. So you can learn more by following this QR code. You can also just kind of search for the publications on the AACR website. We can highlight the link as well. Really worth checking out. It does a really good job of kind of just simplifying and highlighting the different aspects of the CRDC. If you're new or just unfamiliar with all of the various offerings they have, it's a great look at all the resources in an organized manner. So very well written, easy to read. I'm not just saying that because I wrote some of it, but truly it's I think it's useful to learn about the CRDC, right? The resources are already there. So utilize them, learn about them, and work with them to get your analysis completed. So not only the CRDC, but our collaborators were able to publish 9 different publications in 2024. That brings us up to 125 total publications for the CGC. And these are just publications that cite the CGC.
Yeah.
Sorry, go ahead, Sarah.
Nope, that's fine. Continue.
All right, well, we have a list of some collaborators here who have published. If you would like to work with us and be a collaborator, you can check out the slide in just a moment. Here's a listing of all of the research articles that were published this year. Again, we can share the slides so you can have access to links and everything, but just to give you an idea, there's a lot of variety in what was published. Score predicting drug resistance versus an R bioconductor tool. It's really diverse, and that's something I really love about our community is just there's something for everyone and so many different points of view using the CGC to get their work completed. So I really like that. And these are definitely worth checking out. If you are interested in being a collaborator on the CGC or just want to get started, here are some resources for you. You can scan the QR code to be taken to our website where you'll be on that page with information. Luckily, the NCI offers cloud credits to new users. So just simply sign up to the CGC and let us know that you've signed up, and we can deposit that into your account so you can really get started. It's super fast approval, and you can get a lot done with the cloud credits. I think that's like 10,000 hours in R or something like that. So
it's a lot.
It's a lot. And then, of course, we have collaborative projects available if you have a bigger project in mind and need a little bit more than $300 of cloud credits. It's just a simple kind of application you fill out. These are on a rolling basis, so you don't have to worry about, you got to get it in by the end of the year or anything like that. Just follow the links and you can let us know. If you'd like to learn more in person, you can always go to our office hours. I should say virtually, I guess, but it feels like in person sometimes.
It's in person.
You can follow this link or the QR code, of course. We have it twice a week. If you haven't been, you are missing out. We have people come just to say hi sometimes and update us on their progress. We have really deep discussions on Docker and containerizing a workflow. It can be as simple of a question or as complex of an analysis that you need to work through. We're here to support you. It'll be me, Sarah, and 2 of our colleagues as well, who are fantastic and love to be able to assist and really get you going with your analysis, whatever it may be.
Yeah,
sorry, go ahead.
I can't encourage people enough to come to office hours. It may seem like it's something that we're just promoting because we want you to hang out with us, and that's true. But it's also been the case that like when researchers in the CGC community come to office hours, they often end up talking to each other and like sharing ideas and helping each other. So it is becoming more and more of a community time. And not,
I think one of our collaborators mentioned it was the most productive hour in the last 2 weeks.
Yeah.
So, I mean, honestly, it's worth it to come. And if there's something bugging you, you're just not understanding how to proceed with something, or you just need to bounce ideas off of someone.
Yeah.
It's a really great place to have that discussion and kind of understand how to best utilize some of these resources. So feel free to join at any of our office hours sessions, again, twice a week. And with that, I'm going to just say thank you to the CGC community for all of your contributions and making the CGC what it is. Of course, we couldn't do any of this without users of the CGC, right? So whether you're contributing through tools or workflows or just speaking up during office hours. We really do appreciate everything that you are putting forth to make the CGC a friendly and productive resource for researchers.
Thank you so much, Rawan. That was a great presentation of the last year on the CGC. We have just a couple minutes before 3, so I'll stop recording now, and then we can leave space for questions.
Sounds good.
Happy holidays, everybody.
Happy holidays.